Ladies and gentlemen, announcing the arrival of Yang Berhormat Tuan Chang Lee Kang, Ministry of Science, Technology and Innovation MOSTI, and Sir Paulus, Nobel Laureate in Physiology or Medicine 2001, and Chancellor of the University of Bristol, accompanied by our esteemed guests. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh and a very good afternoon. Yang Berhormat Tuan Chang Lee Kang, Minister of Science, Technology and Innovation MOSTI, Sir Paulus, Chancellor, University of Bristol and Nobel Laureate in Physiology or Medicine 2001. Yang berbahagia, Datuk Teknologis Dr. Haji Aminuddin bin Hasim, Secretary General of MOSTI, yang berbahagia, Academician Datuk Dr. Tengku Muhammad Azman Sharifuddin, President of Academy of Sciences Malaysia, ASM, yang berbahagia, Profesor Datuk Wira Dr. Raha binti Abdul Rahim, Chief Executive Officer of National Institutes of Biotechnology Malaysia, NIBM, Mr. Jack Boyer, Chair of Board of Trustees, University of Bristol, Professor Evelyn Welsh, Vice Chancellor, University of Bristol, Academician Distinguished Professor Datuk Dr. Meng, Vice-President of the Academy of Sciences Malaysia, Senior Fellows and Fellows of ASM, representatives from ministries, institutions and organizations, members of the media, distinguished guests, ladies and gentlemen, and also participants who are with us via live stream. Welcome to the Nobel Laureate Lecture by Sir Paulus. Before we move on, let us begin with the recitation of Doa, led by Encik Nur Azwan Hanif Kamaruzaman.
Assalamualaikum warahmatullahi wabarakatuh. A'udzu billahi minasyaitonir rajim. Bismillahirrahmanirrahim. Alhamdulillahi rabbil alamin. Wassalatu wassalamu ala asyrafil anbiya'i wal mursalin wa ala alihi wa sahbihi ajma'in. Allahumma ya Rahmanu ya Rahim. Ya Allah, we express our gratitude to you for allowing us to be gathered here today for the Nobel Laureate Lecture. In the conjunction with this assembly, we ask for your help and guidance. Please make our event go on smoothly. Let this day start with your mercy, continue with your blessing, and end with your forgiveness. We seek your blessing for the flawless progress of this event from the beginning till the end. Ya Allah, kindly give us strength and courage to face all the struggles and gain a wide range of knowledge. Kindly bestow blessings, physical and spiritual health, fitness of mind, peace of mind, and strength of spirit. Allahumma ya Azizu ya Qahhar, Ya Allah, make our country a country that is blessed with everlasting peace and prosperity at all times. Ya Allah, on this occasion, we gather here with hearts filled with hope and offer our prayers for all humankind in the world. May they bless with safety, security, harmony within their land, and bringing about peace for all. ربنا ظلمنا أنفسنا وإن لم تغفر لنا وترحمنا لنكونن من الخاسرين. ربنا آتنا في الدنيا حسنة وفي الآخرة حسنة وقنا عذاب النار وصلى الله على سيدنا محمد وعلى آله وصحبه وسلم والحمد لله رب العالمين.
Thank you, Encik Nor Azwan Hanif. Ladies and gentlemen, to get things started, we would like to invite Yang Berbahagia Academician Datuk Dr. Tengku Muhammad Azman Sharifuddin, President of Academy of Sciences Malaysia and STI Advisor to the Prime Minister and the nation, to deliver his welcoming remarks.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon and salam sejahtera. Yang Berhormat Tuan Chang Lee Kang, Minister of Science, Technology and Innovation. Yang berbahagia Datuk Dr. Haji Aminuddin Hashim, Sekjen of the MOSI. Sir Paul Nurse, Nobel Laureate in Physiology of Medicine 2001 and Chancellor, University of Bristol. Yang berbahagia Profesor Datuk Wira Dr. Raha Abdul Rahim, Chief Executive Officer of National Institutes of Biotechnology in Malaysia. Mr. Jack Boyer, Chair of the Board of Trustees, University of Bristol. Professor Evelyn Welsh, Vice Chancellor, University of Bristol. Academician, Distinguished Professor Datuk Dr. Looi Meng, Vice-President of Academic Sciences, Malaysia. Senior Fellows and Fellows of ASM, representatives from the ministries, institutions, and organizations, members of the media, distinguished guests, ladies and gentlemen. Let me begin by bidding a warm welcome to all participants. Joining us today, we have scientists, researchers, university and school students, and members of the public. I'm sure all of us are eager to know more about the topic of the lecture, what is life? Especially so when the lecture is to be delivered by Nobel Laureate Sir Paul Nurse. Our highest appreciation to Yang Berhormat Tuan Chang Lee Kang for his presence to grace this event. May I express our deepest gratitude to Sir Paul. Coming all the way to Malaysia must be a challenge, given how busy you are with your diverse tasks and responsibilities. I have to admit that I will be listening to the talk as a complete novice. I hope Sir Paul will excuse my ignorance of cellular biology. But we have among the audience people like my colleague, Professor Raha. She is likely to join all the dots immediately upon hearing the keywords that Sir Paul will invoke. Still, regardless of our personal initial conditions, we are all here to learn. It may surprise many that we in Malaysia have a Nobel Laureate program. In the year 2000, then Prime Minister Dr. Mahathir challenged our scientific community to foster a culture of excellence and produce a Nobel Laureate by 2020. That was indeed a stretch target. It is only to be expected that more than 20 years later, we are still on the same journey. What then is a big idea? Simply put, we want to organize our science systems, its people and organizational processes to achieve a higher level of excellence. Learning from the best is a good way to start. Sir Paul Nurse is the 25th Nobel Laureate to visit our shores. The previous 24 laureates include Professor Leland Hartwell, who shared the Nobel with Sir Paul. The Academy has been participating in the Lindau Nobel Laureate meeting since 2004. Thus far, we have sponsored 84 young scientists who spent a few days in Lindau engaging with Nobel laureates and mingling with other young scientists from around the world. We also participate in the CERN Summer Student Program. To date, 23 students have been sent to CERN in Geneva since 2012. Close contact with top scientists has helped to build global knowledge networks that place Malaysia as a credible actor. This may facilitate our younger scientists to enter as strong partners in top flight research programs from around the world. There are great stories to tell, which I will not try to cover here. The little that I know about Sir Paul's work seems to point to the crucial importance of having a systems view of the cell cycle and indeed of all life processes. Control becomes essential, which implies the need to monitor the dominant system dynamics and then invoke some form of feedback mechanisms to keep all the parts operating in harmony. Then the whole becomes more than the sum of the parts. Actually, I'm saying this to do justice to my training as a systems engineer. Sir Paul has spent an entire lifetime obsessed with the cell cycle. He is in the best position to give us new insights on the question of what is life from the scientific point of view. To be sure, the same question has been posed over many millennia by sages and thinkers from all cultures and civilizations. They have given answers from diverse vantage points: philosophical, spiritual, social, economic, and all the way to politics and governance. Given the environmental changes that confront humanity today, it is essential that we endeavor to integrate all of these perspectives into a holistic approach that may lead to a more sustainable future for all forms of life, and thereby safeguard planetary health. Ladies and gentlemen, may we learn much from Sir Paul's lecture and be as obsessed as Sir Paul in pursuing our dreams. Thank you again for the privilege of hosting you in Malaysia. Enjoy the lecture. Thank you.
Thank you Yang Berbahagia Datuk. Ladies and gentlemen, we are honored today to have the Minister of Science, Technology and Innovation Mosti, Yang Berhormat Tuan Chang Lee Kang, grace this event. It is now my great pleasure to invite our guest of honor, Yang Berhormat Tuan Chang Lee Kang, to give an opening remark. Please welcome.
Thank you very much. Yang Berbahagia Datuk Teknologis Dr. Haji Aminuddin Hashim, Secretary General of Ministry of Science, Technology and Innovation, Sir Paul Nurse, Nobel Laureate in and Chancellor of University of Bristol, Yang Berbahagia Academician Datuk Dr. Tengku Muhammad Azman Sharifuddin, President of Academy of Sciences of Malaysia, and Science, Technology and Innovation Advisor to the Prime Minister and the Nation. Yang Berbahagia Professor Datuk Vira Dr. Raha Abdul Rahim, Chief Executive Officer of National Institute of Biotechnology, Malaysia. Very good afternoon. Salam Malaysia Madani. It is with great pleasure and honor for me to open this distinguished event co-organized by the National Institute of Biotechnology Malaysia, NIBM, and the Academy of Sciences Malaysia, ASM, two esteemed agencies under the Ministry of Science, Technology and Innovation, MOSTI. It is a privilege to host a talk by a renowned Nobel Laureate, Sir Paul Nurse, whose contribution to science have profoundly impacted the field of cell biology and beyond. Distinguished guests, ladies and gentlemen, the National Science, Technology and Innovation Policy, NSTIP 2021 to 2030, formulated by MOSTI, is designed to strengthen the role of science, technology, innovation, and economy in the development and growth of an innovation-based economy. STIE aims to innovate and apply science and technology for the benefit of various community levels and the entire country. Regarding the National Biotechnology Policy 2.0, 2022 to 2030, its vision to transform Malaysia into a progressive high-tech bio-innovation nation hinges on cultivating a skilled workforce of science graduates. These graduates are essential for driving research and innovation in biotechnology, meeting industry demands, and advancing Malaysia's position as a leader in sustainable high-tech biotech solutions by 2030. Therefore, the importance of today's talk cannot be emphasized enough. Nobel laureates are not only leaders in their respective disciplines, but also symbols of the pinnacle of human achievement and intellectual pursuit. Sir Paul Nurse's groundbreaking work on cell cycle regulation has not only advanced our understanding of fundamental biological processes, but also paved the way for significant medical, biotechnology, and technological innovations. His relentless pursuit of knowledge, coupled with a passion for discovery, has not only pushed the boundaries of scientific understanding, but has also inspired countless individuals to pursue careers in science, technology, engineering, and mathematics, STEM. It is our fervent hope that Malaysia will also have our very own Nobel laureate from the inspiring work and relentless commitment to enhance our science, technology, and innovation ecosystem. This event also holds significant importance for education enrichment, inspiration and motivation, and strengthening institutional ties. As you engage with today's discussion, I encourage you to reflect on how Sir Paul Nurse's journey can inspire your own research and professional pursuits in advancing biotechnology and many areas in STEM. Ladies and gentlemen, let us take this opportunity to engage with the ideas presented, ask questions, and explore new avenues of thought and collaboration. Thank you to NIBM, ASM, and MOSTI for making this event possible, and to all of you for being here to be part of this momentous occasion. Thanks you all for joining us today. Listening to Sir Paul Nurse will surely ignite a passion for discovery and renew our motivation to contribute to the advancement of science and society. Enjoy the enlightening experience. With that, I now declare the session officially open. Thank you.
Thank you, Yang Berhormat Tuan Chang Lee Kang. Ladies and gentlemen, without any further delay, I would like to call Mr. Jack Boyer, OBE, Chair Board of Trustees, University of Bristol, to introduce our honorable speaker today.
Good afternoon, ladies and gentlemen, distinguished guests, and Minister. It is a great pleasure to be here today to deliver the introductory remarks for this event, which is jointly hosted by NIBM and the Academy of Science Malaysia. We're gathered here for a special occasion, to welcome a remarkable individual whose work has had a significant impact on the field of science and beyond. I'm Jack Boyer, Chair of the Board of Trustees of the University of Bristol. I would like to take this opportunity to also extend my heartfelt gratitude to Datuk Ahmad Zaki Zahid, Chairman of the National Institutes of Biotechnology, Malaysia, and an alumnus of the University of Bristol. He initiated the idea of inviting Sir Paul to deliver a lecture here, but unfortunately he could not join us today as he's still in Mecca. Sir Paul Nurse's contributions to science, particularly in the study of cell biology, have been groundbreaking. He was awarded the Nobel Prize in Physiology or Medicine in 2001 for his work on the discoveries of protein molecules that control the division of cells in the cell cycle. His work has provided a deeper understanding of how cells function and divide, which is crucial for the study of diseases like cancer. Sir Paul's journey in science started with a deep curiosity and a drive to explore the unknown. Over the years, his research has opened new doors in medical and biological sciences, leading to advancements that have a real impact on people's lives. His discoveries have not only expanded our knowledge, but have also paved the way for new treatments and therapies. Amongst many other accolades, he has also received, get ready for the list, the French Légion d'honneur, the Royal Society Copley Medal, the Japanese Order of the Rising Sun, and was knighted in 1999. He also holds honorary degrees from over 70 institutions. In addition to his research, Sir Paul has held many important roles. His leadership positions are almost too many to mention, but notably, he's been a Chief Scientific Advisor of the European Commission, President of the Royal Society, Director General of the Imperial Cancer Research Fund, Chief Executive of Cancer Research UK, President of Rockefeller University in New York, and in 2010 became the first Director and Chief Executive of the Francis Crick Institute in London. So we are privileged to have Sir Paul here to deliver his lecture today. His insights and experiences are sure to inspire us all. Ladies and gentlemen, please join me to give a warm Malaysia welcome to Nobel Laureate Sir Paul Nurse.
Well, Minister, President of the Academy, ladies and gentlemen, what I want to say at the beginning is it is a real pleasure to be here. This is my first official visit to Malaysia. And I am delighted to be here as a President of the Royal Society, a rather older scientific academy. It's a particular pleasure to be associated with the Malaysian Academy itself. So I'm not going to be talking today about my research, or only maybe one or two sentences. I'm going to give a more general talk. What I decided to do was to talk about one of the most fundamental questions in biology. What is life? It's a very easy question to ask. I have to tell you, ladies and gentlemen, it is not so easy to answer. There's an extraordinary diversity of life on our planet. Just look at this extraordinary country teeming with life. All around us is life, bacteria, fungi, plants, animals, and of course, ourselves as well. My talk is about what it is that links all these different life forms together. It's quite a long time ago, but I think it was a butterfly that first started me thinking about questions like this. You have wonderful butterflies here in Malaysia. It was early spring. I was probably 12 years of age, roughly. I was sitting in my garden in northwest London when this quivering yellow butterfly flew over the fence. Well, it wasn't this particular specimen, I must say, but it was a yellow brimstone butterfly, which is the first butterfly of spring in the United Kingdom. It turned, it hovered, it briefly settled, a shadow disturbed it, it took flight again and disappeared over the opposite fence. This intricate, perfectly formed brimstone yellow butterfly made me think, because it was both utterly different to me, yet somehow rather familiar as well. Like me, it was so obviously alive. It could move, it could sense, it could respond. It seemed so full of purpose. I found myself wondering, what does it really mean to be alive? And that is, what is life? Now, the question has been asked by others for many years, centuries. For example, this is the title page of one of a number of books written by different authors called What is Life? This one is probably the most famous. It was written in 1944 by the physicist Erwin Schrödinger, particle physicist. His book was influential, including for myself, when I first read it over 50 years ago. He approached the problem thinking as a physicist. I'm going to take a somewhat different approach. I'm going to consider what I think are 5 of the most important ideas of biology. I'm going to describe where they come from. And I have to tell you, ladies and gentlemen, quite a number of them are quite old. They have been around for quite a number of years. But I'm going to explain where they come from, what they are, and then at the end of my talk, I shall pull them together to develop some principles fundamental to life. But it will not be a neat dictionary-like definition. Just let me assure you, so you don't get disappointed when you get to the end. The first idea I want to talk about is this one, the concept of the cell. The cell is the basic structural and functional unit of all life. I like to think of it as life's atom. Cells were discovered by the polymath Robert Hooke in the 1660s in the city in which I live, in Oxford. We have a plaque on the wall, in fact, recording his discovery. He used a razor to cut slithers off a plant and examined them under an early microscope, similar to the one that you see here. And what he saw, and you see the picture that he drew, were neat arrays of boxes. And these boxes he called cells after the Latin word cella for small cubicle. And these are shown in this black and white picture in a book he published called Micrographia, which Samuel Pepys, who was then president of the Royal Society, said was the most wonderful and magnificent book that he had ever written. I put next to it a picture of a modern scanning electron micrograph of the same material. You'll see our understanding hasn't improved much since the black and white picture drawn by Hooke 3.5 centuries before. A few years later, a Dutch draper working with cloth called Antony van Leeuwenhoek, working in Delft, the city of Delft in the Netherlands, scraped between his teeth and put the contents between his teeth under a microscope, an even simpler microscope than the one described here, shown here, and discovered single-celled life. He discovered bacteria. He sent this drawing to the Royal Society in London, which had only been around for about 20 years. It included the charming images you see here. Particularly, I like the one where we have a figure C, B, C to D, because obviously this bacteria was swimming under the microscope and doing little loop-to-loops. I have to tell you, in the letter which he sent to the Royal Society, which I have read, and I've handled this particular letter here, because it's in the archives, he said he was very disturbed by the discovery of living organisms in his teeth, because he was very proud of his dental hygiene. And to find life in his teeth, he thought was very disturbing. Now, he was a close neighbor in Delft to the famous painter Vermeer, who painted, unusually, 2 pictures of a scientist. This is one of them. One is in Frankfurt, the other is in the Louvre in Paris. One was an astronomer, the other was a geographer. It was the same person. I like to think this is Lowenhoek. They were neighbors. But I admit to you, there is no good evidence for this speculation, except I like the idea. And when Vermeer died, which he died at a young age in his 40s, Lowenhoek was the executor of his will. So it is possible that they knew each other, and I think it's possible he had an idea, a concept of a scientist through Lowenhoek that he painted here. Over the next 2 centuries, it became clear that all living things consist either of a single cell or a collection of cells like ourselves. This conclusion was summarized by the German zoologist Theodor Schwamm in 1839 in German, translated here, where he said, "We have seen that all organisms are composed of essentially like parts, namely of cells." This is, of course, the cell theory, the structural unit of life. But as well as that, they are also the basic functional unit of life. This was well expressed by another German scientist, actually the founder of pathology, Rudolf Virchow. He, in German, of course, this is a translation, stated, 1858, "Every animal appears as a sum of vital units, each of which bears in itself the complete characteristics of life." What he's saying there is cells exhibit the properties of life. And that means that they are important in thinking about what life is. He also said, "Omnis cellula e cellula," Latin, "all cells come from cells." It wasn't clear at the time he was writing whether they somehow emerged from murky solutions. But in fact, and I shall refer to it at the end of my talk, all cells are formed from previously existing cells. So cells are interesting. They're very interesting. But if I've failed to convince you of that, let me show you this picture. This is a picture of a mammalian cell being fertilized by sperm. And I want to remind all of you in this room, all of you once looked just like this. You were all once a single cell. And if I fail to interest you, there's a couple of doors out the back that you can leave now. Cells are considered to be the simplest entities which unambiguously exhibit the characteristics of life. We have to deal with viruses a bit later, but for the moment, unambiguously, cells are the simplest entities you can say are alive. That means understanding cells is key to understanding life. So it's going to be central in what I have to say. This is a another modern picture of a cell. They are bounded physical entities. They're separated from their environment. They're surrounded by a semi-permeable membrane, a lipid membrane, which separates the cells from their surroundings, but allows communication with this surroundings. I want to emphasize that separation because it allows order to be built up inside cells at the expense of increasing disorder outside cells. In this way, life does not contravene the second law of thermodynamics, and this is important because it's a law that often troubles physicists thinking about life. Like Schrödinger, as seen in this quote from 2 quotes here from his books. I like this sentence. He was astonished. An organism's astonishing gift of controlling a stream of order on itself and thus escaping the decay into atomic chaos. Great sentence, great sentence. He also had a second sentence I quote here. He referred to chromosomes. Chromosomes in cells contain in some kind of code script for the entire pattern of the individual's future development and function. These quotes introduce the second idea I want to share with you, and that is the idea of the gene which can explain the mysterious phenomenon of inheritance. This is the idea of the gene as the basis of heredity, and it had its origins with the work of an Augustinian monk, Gregor Mendel, abbot of Brno Monastery, working in the 1860s on a research project I'm going to tell you about. What he did, he tackled the problem of inheritance by carrying out crosses between pea plants with different characteristics, and analyzing the outcomes in terms of the thousands of progeny formed in these crosses. This is a picture of his garden. I visited it in 1981, visited the monastery. It was the height of the Cold War at this time. Very difficult, or not easy, to get here. What I want to emphasize to you, this was quite a large garden. It was a research garden for doing pea plant crosses. He also had a greenhouse of similar size. This monastery was essentially a research institute interested in science because it was a teaching order. Mendel himself studied meteorology, monitoring the pressure of air as storms went through. He did a map of the moon, but he also carried out these crosses of plants. And he chose peas for several reasons, but a main one was that there were a range of pea varieties with different characteristics, character types. We call them phenotypes, tall plants, short plants, different colors of flowers, different seed shapes, and the like. This meant he could monitor the characteristics as you go through different breeding cycles, because if you can't describe them properly, you can't really you can't really follow what's going on. And he famously observed simple ratios in these characteristics in the progeny produced by the crosses he was carrying out. I'm not going to explain Mendelian genetics to you, it will take me too long, but ratios like 3:1, and 9:3 to 3:1, and 1:2 to 1. And this led eventually to the idea of Mendelism, which was the idea, it's essentially a particulate idea, which is the reason you get these simple ratios, because there's particles being segregated here. And he had the Mendelism is the idea that heredity is based on pairs of indivisible particles or factors which are inherited from those germ cells, the pollen and the ovules of plants, sperm, and eggs in animals. These factors were subsequently found to be linked to the chromosomes, which Schrödinger mentioned many years later. These were not discovered until 20, 30 years after Mendel's death. Mendel's work had no impact at all at the time. It was not until it was read 35 years later that it became important. And what happened between Mendel and his rediscovery was the discovery of chromosomes. This is a picture taken in 1890, or drawn in 1890, I should say, of chromosomes separating apart as cells divided. And what scientists began to realize is the factors that Mendel described hypothetically just by counting numbers, could be represented in the concrete description of the chromosomal behavior. And it moved from being an abstraction to a concrete behavior. And that's what I think made the whole thing more convincing. It wasn't just an abstraction, it was also experienced as reality. These factors are, of course, what we now call genes. In the 1940s, Oswald Avery in Rockefeller University, where I worked for 7 or 8 years in New York, showed that the chromosomes were made up of deoxyribonucleic acid, or DNA, 1944. Most scientists didn't believe it at the time. It took another 10 years to convince them. And that's when the structure of DNA was established about 10 years later in London by Rosalind Franklin, Ryan Gosling, and Maurice Wilkins, and interpreted in Cambridge, UK, by Francis Crick and Jim Watson. The Crick I named the Francis Crick Institute after, because probably our most famous life scientists in the UK in the 20th century. Crick and Watson famously argued that the double helical structure of DNA could explain heredity through the linear sequence of bases that you can see here making up DNA and the pairing between bases, adenine pairing with thymine and cytosine with guanine. Look at this. This is a more abstracted picture, of the double helix undergoing copying, undergoing replication. The double helix is a twisted ladder. Imagine a ladder and twist it. But to think about it, untwist it in your mind, okay? Untwist it back into a ladder, and you have a ladder with rungs. And the rungs are made up of pairing between bases, chemicals, in each of the side sections of the ladder, where adenine, or A, pairs with thymine, or T, and guanine, G, pairs with cytosine, C. And what this means, of course, is that if you now separate these rungs, break them and separate them, they provide a precise template to build another identical molecule. So you get 2 identical molecules on after the replication. This explains how DNA is copied, accounting for heredity. It explains Schrodinger's code script and the permanence of the gene between generations, because DNA acts as a code script which is precisely copied every time a cell divides. Beautiful. Crick, who was quite a smart man, I have to say, I met him a number of times, brilliant in fact, went on to propose what he called the central dogma. He wasn't backward in his confidence either, I have to say. So he proposed the central dogma, explaining how DNA encodes the structure of proteins. That's shown here. It shows how the sequence of bases which make up a gene is copied first of all from DNA to RNA, ribonucleic acid. That acts as a messenger from the nucleus into the cytoplasm. In the cytoplasm, proteins are made, which are determined by the code of DNA that's been copied into RNA, with generally one gene encoding one protein. These were fundamental discoveries. They lay the foundations for molecular biology, for biotechnology, for advances in medicine. They are all extremely important, and they are critical also for understanding life. Central to this is the genetic code, written in 4 letters, the 4 bases, with the information stored in a linear form. Let me emphasize that. I'll return to it, in a linear form. This way of storing information is the same as the sentences I'm speaking to you. They are linear. Or the sentences you read in a book, that's linear. And it tells us that linear digital storage of information was invented by life over 3 billion years before the computer age. Puts us in perspective. 3 billion years ago, life had it. Now we have computers that have it, but we didn't think it up first. Life did. Well, we're alive, of course, but, you know. Okay. The nature and function of the gene introduces the two next ideas I want to mention to you: life as chemistry and life as information, which our president I'm going to refer to systems a little later. Now, life as chemistry, that is, the functioning of living things is based on chemical activity and physical forces. The origins of this idea can be traced back to Lavoisier, the father of chemistry, essentially. Unfortunately, he lost his head in the French Revolution. He was guillotined because he was a tax collector. Not a very good one, but he was a tax collector. And although he supported the revolution, by the time Robespierre had gone out of control, he lost his head, and that terminated his research project, not because he didn't have the funding, but because he was dispatched with the loss of his head. But it was another French chemist, Louis Pasteur, 50 years later, who carried out experiments on sugar beet fermentation, an applied project, which led more thoroughly to the concept of that life is chemistry. This is Louis Pasteur, rather romantic picture in the Pasteur Institute. And what he was doing was analyzing the fermentation of sugar beet to make alcohol. And he was doing it at the request of industrialists because the fermentation sometimes failed, and instead of making alcohol, they made acid instead. So they wasted their money. Sugar beet was down the tube, down the drain. And he was asked to investigate it, an applied project. But it came not only solving their problem, but led to a fundamental discovery. He demonstrated that the successful production of alcohol required the presence of a fermenting organism, a single-celled one called yeast. And he proposed that the yeast cell produced substances that made alcohol from the sugar in the sugar beet. And if the yeast was not there, then bacteria would infect. And the bacteria did a different sort of chemical changes which made acid instead, and that was a waste of money. They didn't want acid, they wanted alcohol. So Pasteur solved the problem for the industrialists. I don't know if he was paid, I've not found that out, or whether he was just helping the local society. But he concluded more generally that fermentation was a physiological act yielding products for the cell. That is, chemical reactions are an expression of the life of a cell. That's a direct quote from him in French. In other words, life is chemistry, but it is a very elaborate and a very special sort of chemistry. It's based on carbon polymers, as is DNA, RNA, but now we have to consider the carbon polymers that make up proteins. These form the substances from yeast that Pasteur postulated made the alcohol. They are enzymes which catalyze chemical reactions. Now, proteins are polymers of amino acids with a backbone and side chains of amino acids. Here we see the backbone. The backbone here is the nitrogen and the carbon atoms all connected. But the different amino acids have different side chains, and they're chemically diverse. I want you to remember that, chemically diverse, positively charged, negatively charged. Sometimes they repulse water, some are bulky, some are small. We have 20 amino acids, as does all life, or nearly all life, that make up these proteins. This is another picture emphasizing the atoms which make up DNA. Now, the presence of very varied chemistry in proteins is in complete contrast with DNA. DNA is stable, it's not chemically diverse. In fact, it's a rather dull molecule. And if you want to store information, you want it to be dull because you don't want it to change. But it can't do chemistry. Proteins can do chemistry because it's made up of all these different chemical components. And the chains of amino acids, this is a typically small protein, a couple of hundred amino acids, but their different chemistries mean They can fold up in different ways, make different shapes. You can see this a billiard ball structure. The different colors indicate different chemistries that are linked to different parts of the protein. And what this means is that a wide diversity of chemical reactions can be carried out by enzymes, because you can create very different structures. You can't do that with DNA, and the structures are chemically diverse. And these proteins act as chemical machines. They produce the huge range of chemicals required for living organisms to function. We talked about the biotechnology briefly at the very beginning of my talk. I think I mentioned it. It's capable of undergoing all sorts of chemical reactions under the mildest conditions that we humans may have to heat to high temperatures and high concentrations and high or low pHs. Cells do this under much milder and simpler conditions. That's one of the reasons they need to be thought carefully about with respect to biotechnology. They can make new molecules, they can break molecules down, they can generate large molecular assemblies, they can make all the components that make up cells. They can also do physical work. They make motors that run around on railway tracks inside cells, minuscule railway tracks. They can capture light from the sun and turn it into energy-rich chemicals. This is a mixture of physics with chemistry. Now, and this is important, all this activity means that there's thousands of chemical reactions being carried out simultaneously and close to each other in the tiny spaces of living cells. This is why it's so elaborate and so complex. A small sample of the reactions of life is shown here. When I was a student many years ago, I had to remember what every one of these dots meant. I wasn't very good at examinations, and this is one of the reasons I could never remember all the dots. And luckily we've moved on now because we have Mr. Google, and you don't have to remember all the names of the dots. You need to think more, what does it mean, rather than describing what it is. But that's a different lecture, I have to say. So, why I mention this is that all these reactions are going on simultaneously, but for it to work, they have to be separated one from another, because they each have a different chemical microenvironment. And that separation is brought about by the surfaces and folds of the enzymes that can isolate the chemistries one from another, by complexes of different proteins linking together, or by membrane-bounded organelles visible back to that cell. We shouldn't just think of this as a structure. We should think of this as a complex set of chemical microenvironments which are differing one from another to allow all this chemistry to take place. Here's a schematized picture I probably stole from Scientific American or somewhere like that, that shows the different chemical reactions that are going on in these different compartments. And this allows the huge, elaborate chemistry of life to take place. However, for this to work properly, the chemistry has to work together as a whole, as a system. You can't just have random chemical reactions occurring. They've got to be connected. This connection requires communication between these different microenvironments, communication between the compartments. And the transfer of information, which is the basis of communication, leads to my next idea, life as information. Complex systems, control, purpose, where we can learn something from the engineers, as you are about to see. Life is a complex system. I wanted to find out who first started talking about this. You will be as surprised as I was when I found we could trace it back to the philosopher Immanuel Kant. He wrote a book on moral philosophy and has about 20 pages talking about life as a complex system. What it has to do with moral philosophy, I have no idea, but it is certainly the first account I have found that argued this. The operations of the complex systems of life are based on the management of information. And I'm going to give you two examples to illustrate this. The first is the structure of DNA. You're not going to be surprised I refer to this. Now, the structure of DNA, the double helix, is iconic, it is beautiful, though it is overdone. When I set up the Francis Crick Institute, I said, I'm having no statues of a double helix in it, because every damn life sciences institute has got double helices in it. I'm sick of them, okay? I discovered a few weeks ago this one appeared somewhere on one of the upper floors. I don't know why, I don't know who put it up there, but it is up there. I shall deal with it when I return. So it is beautiful, but its real beauty lies in the fact that it only makes biological sense when it is recognized as this digital information storage device. That's the real beauty of DNA. That's what it is. One written in this linear script of 4 letters. Now, you can see the DNA, the coded elements are turned inside the molecule, protected, in fact, and they're chemically inert, which is exactly what we want. But this leads to one of the really important conclusions. I haven't really read people emphasizing this, and I want to draw your attention to it, because the molecule is stable, DNA, but it can be turned into chemical action that can do work through the proteins. So you have stable chemistry, unstable chemistry, which you need to actually do work. This is a wonderful characteristic of life. It can code information and it can do work. And they have to be separated, because if they're not separated, you'll confuse it. It's based on carbon-based polymers, the ability to encode information into inert one-dimensional structures and carry out chemical and physical work in active 3-dimensional structures. So that's one example. The next example is about regulation. That is, making complex systems in living organisms act as a whole with purpose. Now, I don't know if you're like me. I understand most machines built before 1900 and increasingly fewer machines built after 1900. This is a governor on a steam engine which James Watt developed, from Huygens actually, who used it in windmills, but 100 years before. And it helps if we look at man-made machines as metaphors and analogies for thinking about life. This governor is easy to understand. It goes round, driven by the steam engine. Those weights swing out. It's intuitive, yes? It lifts a valve, shutting off steam going into the engine. The spindle stops rotating as fast. The balls go back in, it lets in the steam. It's a perfect homeostatic mechanism. It's an example of a negative feedback loop, which I show you here, up the top there. This is now back to a living thing. We've got a chemical A to B to C, catalyzed by the green arrows. When C builds up, it inhibits A to B. It's a negative feedback. Beneath it, you see a positive feedback as A to B to C, but as C builds up, it promotes A to B. It forms an irreversible switch. Now, other control models exist which operate in cells. They generate switches and timers and toggles and oscillators, all translating chemistry into informational modules. And these can be linked together to make more elaborate ways of informational management and processing. We have things, by the way, to learn from living organisms and how they operate, which will help us in the management of our complex machines and probably also machine learning and artificial intelligence. A metaphor for all of this is the electronic circuit. This is a transistor radio, I think. Again, I stole it from somewhere. And what it was an EMBO journal, that's right, a journal. And what they had done, who wrote it, they'd called different elements, different oncogenes, genes that cause cancer. And the idea is they're all linked together to produce the behavior of the cell, which when it goes wrong, can generate cancer. Now, this, by the way, is a hardware circuit. And it has been argued that living things, you're best thinking of it as a wetware circuit, because we connect the different components through solution chemistry, which means you can reconnect into different places, much more versatile, not yet properly investigated, something people should look at. Now, this allows the system, it's complex, but to behave as a whole with overall purpose, such as reproducing a cell, making a butterfly, maintaining our cells. These are purposeful behaviors. Now, for centuries, the generation of purposeful behaviors was thought to require a designer, somebody who could design it. And it took evolution by natural selection to change this. And this is my final idea. If you're getting tired, I'm getting near the end of my talk, okay? So deep breaths, we're getting there. Deep breaths. Now, evolution by natural selection, how living things can come about. Now, this beautiful idea is mainly associated with Charles Darwin, but also with Wallace, who, of course, did many of his investigations here in the Malayan archipelago and further east. The idea has two parts. The first is that life evolves over time. The second is a major mechanism for this is evolution by natural selection. Now, the first of these ideas had been discussed for 100 years before Charles Darwin. I'm going to give you one example here, which is Charles's grandfather. Charles is the rather serious gentleman on the right. His grandfather, Erasmus Darwin, is perhaps slightly less serious looking person, I would say, on the left. I want to say a few words about Erasmus Darwin, because he's nowhere near as well known, and he's an interesting character. Colourful. He was a scientist, he's a doctor, but he was a poet. And he wrote all his scientific observations and conclusions up in the form of poetry. I have collected his books, were published in the 18th century. They're written in poetry. He was a Republican. He refused to be the doctor of George III, and he was asked several times. He was a proponent of public women's education, and he wrote a book about what girls should be taught in public schools, and he opened a school that was taught by his two daughters. And he was a great conversationalist, despite a terrible stammer. And by the way, he lived in the Midlands in England, north of Birmingham toward Derby, and there wasn't a university within 100 kilometres of what he was doing, completely independent. Now, the evolution, so that was Erasmus. He talked about the evolution of life, so did others like Lamarck and so on. And the evolution of life, the idea that it evolves over time, implies there's a tree of life. That is, that living organisms are related one with another. And if life only arose and survived once, and remember it has the same chemistry, so that's likely, then all life on Earth is related to each other. And this is captured with the only diagram in Darwin's Origin of Species, published in 1859. You're noticing how most of what I'm telling you about was initially discussed before 1900. And this is the first tree of life, as you can see there. Now, Charles Darwin gathered evidence in favor of life evolving through natural selection during a trip around the world that took 5 years in a boat called the Beagle, which was a small warship of the British Navy. Let me briefly explain natural selection. It will be brief. Natural selection is the consequence of a population of species which exhibits variations, which are caused by inheritable genetic changes. These can be caused by mistakes when DNA is copied or if it's damaged. Now, if these variants influence characteristics that make organisms more successful, better adapted, and therefore reproduce more, then that particular genetic variation will spread through the population. It's such a simple idea, but it's a beautiful idea. Over time, such genetic variations accumulate, they lead to changes in the population to the extent that individuals of a new population eventually may be no longer able to breed with the original population, leading to a new species, which accounts for the title of his book, The Origin of Species. That's what interested him about it. He came to this view in part by his studies of the beaks of finches in the Galapagos Islands in the Pacific. Now, once again, I didn't draw this, I took it from somewhere. On the left, you see the finches. On the right, we have pliers, okay? The finches are modified to do things that finches want to do if they are specialized. The one at the top can break nuts. You can see how it would break nuts. The one at the bottom can get under the bark of trees and catch insects. This is just like pliers, which are variants on a theme, but they're better for doing different jobs, as any mechanical engineer or electrical engineer in the room would realize. The difference here is, of course, one is formed by natural selection, the other one designed by the different engineer. Now, for natural selection to take place, three things are needed. Reproduction, obviously, a heredity system, obviously, DNA, and that heredity system needs to exhibit variability upon which selection can work. And these are based on the ideas of the cell and the gene, which I've just explained to you. Imagine a single celled life form with a brown coat, or is it orange up here? It might be orange looking, okay? Imagine that there's a gene that determines the color of the coat. Imagine that if it's mutated, it can make a red coat. Also imagine that the red-coated cells survive better. Maybe they avoid predators or something. And as a consequence, the red cells replace the brown cells in the population, and we, though, evolution by natural selection has taken place. That's what it is. That's a simple explanation of it. But it is profoundly important. So much so that in the 20th century geneticist Hermann Müller used it to define what life is. He said, living things have properties which allow them to undergo natural selection and to evolve. And I'm going to return to that in the closing remarks of my talk. But before I do that, I want to say goodbye to Charles Darwin. He's got older, he's got a bigger beard, as you can see here, and I'm going to read you the monumental sentences that end his Origin of Species in 1859. Whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. I wish we could write scientific papers like this today. I can't bear to read my own papers a year after I've written them. By the time they've been edited and gone through reviews and so on, anything of any interest, of any color, or of any personal nature has been edited out. You can still read The Origin of Species and be excited by what you actually read. Also note here, because he was a subtle person, he refers to gravity. Bit odd, you know? And I think he refers to gravity because Isaac Newton was the king of physics, and Isaac Newton had laws of physics, and I think Darwin wanted the reader to think there are laws of biology as well as laws for physics. Again, I have no evidence for it, but I speculate, because it's a strange sentence. Now, I'm about to try and define life, but I need to consider viruses, because somebody will ask me about viruses. Are they alive or not? Viruses have a genome based on DNA or RNA. They undergo evolution by natural selection, so they pass Muller's test, but they can only reproduce when they are in the cells of other living organisms by hijacking that machinery. And this is the reason people worry about it. Now, I'm going to give you a different view of this. I'm going to remind you that actually it's important to remember that all forms of life are, to a greater or lesser extent, dependent upon other living beings. We tend not to think about that. But of course, we're completely dependent. We eat plants. We eat animals. We can't make all the amino acids we should make. And that is the same, essentially, for nearly every living organism. I can't think of one which is really, it's not like that. Parasites that live on or inside the cells of bodies of animals and plants are an example. The point is, the level of dependency changes, but the principle doesn't. We are all dependent to some extent on other living things. Once you've realized that, then you realize the way to think of this is as a graded spectrum of dependency. Different life forms have varied dependencies. In the case of the virus, it's very strong. In the case of other living organisms, it can be much weaker. But I would argue that they are actually similar in being dependent upon other living things. So let's not get too excited about viruses. I mean, I know the world of human beings is made up of people who like to split things up into little compartments and people who like to merge them together, but I don't think it's fundamental. But what is fundamental is that life on Earth is fundamentally connected, both through evolution and also through deep relationships and interdependencies. Okay, I'm now going to sum up and then say a few words after it, okay? I'm going to sum up with these slides of principles. Living things are independent, bounded physical entities. That's so I don't have to discuss with anybody computer games, okay, which are not independent physical things. They are separated from their environment, but communication with their environment, they therefore can generate order and organization. The basic unit of life on Earth is the cell, and multicellular organisms are aggregates of cells. Second slide. The central principle for defining life, I would say, is Muller's. Life forms are entities that can undergo evolution by natural selection. Through this, they acquire purpose, better adaptation. They are entities that build, maintain, and reproduce themselves, and they can evolve because they reproduce. They have a hereditary system. It exhibits variability. Life forms are chemical, physical, and informational machines. They construct their own metabolism. It's used to build and maintain themselves, to grow and to reproduce. This is why I said it wouldn't be like a dictionary definition. You can imagine how much space this would take up in the Oxford English Dictionary. But it is a set of attributes that we need to think about in thinking about life. I'm going to add a few more, which are more specific to life on Earth, which is based on carbon polymer chemistry with information storage devices made of nucleic acids, proteins based on chemical and physical machines, integration together requiring the management of information, and living things on Earth are related by common descent and closely interconnected. Now, should we ever encounter life elsewhere in the universe? Encounter, that means we have to encounter it. I mean, if it's a long way away, it may be difficult to encounter it. But I think, although the chemical details may differ, the principles which I've described here, I think will apply. And I also think that the need for long-term information storage and also for varied chemistry is most easily solved by building it on polymers. So I think it'll be a polymer-built living thing. I'm speculating, of course. But I'm going to finish by returning to our planet, because this is the only corner of the universe where we know for certain life exists. The life we are part of here is complex. Understanding it is fundamental to our culture, to our civilization. It can improve the lot of humankind. But the knowledge goes further. It shows us that all living things we know are related and closely interacting, and we are bound by a deep connectedness to all other life, including the yellow brimstone butterfly that I started my talk with. Together, all of these species are life's great survivors. It's the way to think about it. They survived evolution. They're still with us. Now, this is a critical sentence. I want you to listen to this sentence, because these are the latest descendants, including ourselves, of a single, immeasurably vast family lineage that stretches back through deep time as an unbroken chain of cell divisions. Unbroken chain of cell divisions. Think about it. We are made by repeated cell divisions. We were formed by the gametes from our mother and father, formed by cell divisions. And so it goes on and on and on back through time until we get to essentially the origin of life. We are still connected to that origin of life 3.5 billion years ago by repeated rounds of cell divisions. It's extraordinary, and I've never thought of it like that until I started thinking about this. The other thing I want to end with, as far as we know, we're the only life forms who can see this deep connectivity and reflect on what it might mean, and there's different ways you can reflect on it. But one thing I would argue is this. I argue that it gives us human beings a special responsibility for life on this planet, made up as it is by our relatives, some close, some distant. We need to care about it, we need to care for it, and to do that properly, we need to understand it. That's why I've given you this talk, and that's why I've written a book, should you be interested. It's in many languages, and you can hear more about what I've said. Thank you very much for listening to me. Thank you.
Thank you, Sir Paulus, for the insightful talk. Ladies and gentlemen, please join me welcoming the Vice President of ASM, Academician Professor Datuk Looi Meng, who will moderate the Q&A session.
Thank you very much. Won't you agree with me that this was really a very heartwarming lecture? At least for me it was, and I really, really enjoyed it, Sir Paul, because, well, I'm a pathologist and I look at cells. And it is very meaningful what you have said to explain the complexities of the cell and of life and relate it in such a personal way to what we are and how we function. So I'm sure there's a spellbound audience here who have listened to you and they would have many questions. But I'm going to take the privilege of asking the first one to set the ball rolling. And I ask this with a personal agenda. Not so personal, but because I'm a medical doctor and a practicing clinician, can I ask you, listening to what you say, can people really say that DNA is your destiny? The way I've described it, your life is controlled by the DNA, and that transcribes to RNA, to proteins, and make what you are. But as doctors, we always tell patients that you can behave yourself better and do better, and you don't always blame everything on your genes, on your mother, your father, your grandmother, your grandfather, and all these millions of years of inheritance that you have. Can you share with us some thoughts about perhaps epigenetics and how that can modify the expression of your genes so you can be a better and healthier person?
So thank you for the question. DNA is very important, of course, for heredity. Epigenetics in its most general sense, which was, it's a term that was set up by Conrad Howell Waddington in the 1960s, somebody I met actually when I was a student and a postdoc in Edinburgh. And he was concerned essentially with the changes of gene expression that come about in formation of a living organism, which is made of cells which have got different expression patterns. Now, the term epigenetics, it describes all those sorts of changes. And actually, many of us study exactly those changes. But the interest in epigenetics in the popular mind has got a little more restricted into inheritance, particularly between organisms, generations. And there, I think it's probably been exaggerated in the amount that happens. So I think the notion of epigenetics, that is regulated gene transcription to give differing behaviors, is absolutely central. But the evidence for inheritance although present in some simple organisms, in humans there's only very limited evidence that it may have an effect. The reason it is so talked about is because I think popular authors can get their books published by talking a lot about it, and because it seems to challenge, a basic principle of genetics and based on DNA. I'm more inclusive. I think epigenetics, looking at the expression changes, is critical, but I think we shouldn't exaggerate it too much in terms of inheritance. I study epigenetics if we look at changed expression, but actually I wouldn't be seen as an epigeneticist because it's more specific. Now, I don't know, I'm coming back to you. Do you think I've answered your question, or do you think you would like to come back on it?
You have answered my question. Thank you, Sir Paul. As I told you, I was trying to recruit you to try and to convince the audience that they shouldn't totally blame their genes for who they are, for their hypertension and for being obese and so on. They could modify their behavior to some extent.
Can I say something about that, actually,
yes.
The modification of behavior? Because we rapidly get into discussions about free will, and I'm not a philosopher and I'm not a humanist. There's others who are in the room who are better trained at this. And it's a complicated topic, of course, because people say, well, if I'm fully determined, I can't take responsibility for action and so on. The only thing I wanted to bring to your attention here is that we are a consequence of our history as an organism, our experiences in life. And those experiences in life lead to changes and differences in the decisions that we make. So we can make decisions based on our experiences, and that is still perfectly compatible with a deterministic view, and we shouldn't get, again, too agitated about that. But it is a complicated issue, and I am absolutely not trained to talk about it, so I'll lead it to the philosophers.
Thank you very much. And I shouldn't dominate the conversation anymore. I'll be very happy if I'm sure there are questions from the audience. There are microphones.
I should say I'm a bit deaf, so I may need to have it repeated for me. Okay. Is there a microphone?
There is a microphone.
It's coming.
So,
hello.
Yes, Prof. Yasmin. I think good to say who you are, where you come from in a few seconds, so we conserve the time.
I'm Yasmin. I'm sometimes a virologist, but today I'm going to ask you a question. Going back to this idea of viruses, I know a lot of people dismiss viruses as not part of the evolution of life. My question is, what do you think of the idea that, I agree viruses are parasites, but on the other hand, in the food chain or in the cycle of life, viruses itself nothing depends on viruses in this earth. And yet they are there. And we've just come out of an epidemic or pandemic. Is it possible that these little bits of information are really the agents of change in the big picture of evolution? That when we come out of a pandemic, that we are substantially changed as humans? I mean,
yes.
In other words, if I may summarize it, it would be like perhaps viruses have a purpose too, because as it seems, viruses are put aside as just bits of DNA. They're doing all kinds of things, but could there be a bigger purpose in viruses among us?
I would rate viruses as alive, for sure, and I think they function with purpose. So the fact that they're completely dependent upon another living thing inside a cell, for me, isn't the key factor, because, as I said, to a greater or lesser extent, we're all dependent upon other living things. But of course, it behaves with purpose, to reproduce itself, and that's why it exhibits variability, and that's why it undergoes natural selection. So I'm with you as a virologist. They're alive, they have purpose, they're very interesting, and not only that, they're really, really important to understand, as we have seen with COVID, and as we saw in this region with SARS also 20 years before. But they behave with purpose. I agree with you.
Thank you. Who's the mic going to next? Prof. Raymond.
Hello.
Hello, I'm a quantum physicist.
Ah,
I have two questions. First is, what is the purpose of the non-coding DNA? I would like to know what is the purpose of
let's
this.
do one question at a time. Do you mind?
What is the purpose of the non-coding DNA?
The non-coding DNA is, I would view it as having largely an epigenetic function. That is to say, one which regulates the coding genes. It's mainly regulatory. For a long time, it was thought to be just junk that didn't do very much. In the organism I study, yeast, there's very little non-coding DNA that has evolved with more complex organisms because it is a more complex form of regulation. So I just think it's another example of the extraordinary management of information and management of systems that has now started to use the RNAs encoded by the DNA that isn't encoding proteins, which has led to a very elaborate control. And it is striking that, for example, our own DNA, only a few % of it encodes proteins, and the rest of it was thought to be junk, but isn't junk. It is actually regulating. Okay, second question.
Thank you for the answer. So my second question is, what do you think is the how do you explain conscious life? How do you determine that the life is conscious from the maybe molecular biology perspective? At what stage we can determine that the life has a consciousness and it has a free will? Yeah,
well, I should have I got that one. You're a quantum physicist, so you like difficult questions, yeah? And this is a difficult question. I avoided talking about consciousness in my talk, and I avoided it. I tell you why. I avoided also talking about the origin of life too, because I wanted to write a book and give a talk which might be a bit boring, but actually would be largely right when you read it in 50 years, okay? I think nearly everything I told you is likely to be right. Whereas most popular books and popular talks have stuff, half of it won't be right next year or 5 years later. So they will speculate on consciousness. So if you want a sensible view of consciousness, there's plenty of books in the library. If you think that they are going to last, I'm not so sure about that. Now, having said that, what can we say about consciousness? And this is it's a philosophical question for me, really. I mean, psychological and philosophical. We feel we have consciousness. We worry whether animals have consciousness, and some worry whether plants have consciousness. Depends quite how you sort of define consciousness, but some thinking about it is rather embracing, and it almost could be used to embrace all living things, in which case it sort of starts to get close to purposeful behaviors, to be quite honest, and they may be related. But let's try and be practical. Consciousness as we sort of think of ourselves. And I have a feeling that type of consciousness is a quite highly evolved property and is associated with our extraordinary brains that has resulted in us being effective in all the sorts of things that we do if we embrace the idea that we are conscious human beings. In other words, I suspect that consciousness may have something to do with the fact that it makes us more effective in some way if we think we are conscious. This is a very I don't want you to take me too seriously, but that is how I would sort of start thinking about it. But really, this is very, very difficult. That and free will, origin of life, tricky, tricky subjects.
Thank you. And it also is really right into philosophy as to...
And it tells us, actually, that there are real advances in our understanding that can come about by conversations and discussions between the natural scientists and the humanism and the culture to lead to tackling these problems. For sure, scientists can't tackle it alone, and we have much to learn from these other disciplines. I strongly encourage those sorts of interactions. I'm not entirely confident that we will solve the problems, but I think we should try, and it has to be very interdisciplinary and multidisciplinary.
Thank you very much. Are there any... yes, there's a hand there. Please introduce yourself.
Good afternoon, Sir Paul.
This part of the audience is very productive.
My name is Nik Zakia. I'm from Chempaka International School.
Hello, yeah.
Hello. I have a very simple question, which is related to the statement, we eat to live. We eat to live. Okay.
She's saying that her question relates to the statement, we eat to live.
Okay.
Yes, we
when
eat to live.
When
Yes.
we teach biology to school children, the characteristic of life, we even have a mnemonic to describe characteristic of life, which is Mrs. Grain. Living thing will move, respire, they are sensitive to surrounding, they grow, they reproduce, they excrete, and also they need nutrition. Most of the mnemonic that we give to our students, Sir did mention. So just that I'm a bit curious why there's no explicit mention of needing nutrition. So perhaps, but I think respire could be one of the chemical reaction taking place, but needing nutrition, perhaps that's why when we teach students, we always say viruses are non-living things because of course they don't need nutrition.
Yes.
And yeah,
So
thank you so much.
I think I got it. I
Thanks.
did actually allude to it. I said in the section when I'm talking about chemistry, I'm talking about the changes that have to be carried out in the body of different chemicals. And I also said briefly, but only briefly, that of course we rely on eating and across the board. But I didn't mention the word nutrition. You're absolutely right. Now, I think an interesting question here, about which I am not an expert, is knowing what is proper to eat nutritionally and what is good for us, if you like. And the reason I bring it up is in evolutionary terms, because we evolved in a different way of eating and agriculture. In fact, no agriculture at all. We were hunter-gatherers until 20,000 years ago. So we just lived off what we could catch, animals, what we could collect from wild plants. It was, of course, the generation of agriculture of around 10,000 years ago, which had dramatic changes in our diet. Now, I'm interested in this because we now have the capability of tracking the genetic changes in human beings that occurred as a consequence of the changes in diet, of nutrition, that happened in the Fertile Crescent with wheat and cereal, and then rice elsewhere, and maize and so on, which caused dramatic changes. You as a nutritionist, I'm sure, would agree with it. But we can trace the genetic changes that happened in human beings because we now can sequence the DNA of individuals from 20,000 years ago, 40,000 years ago, and then as the agricultural revolution occurred, and for that matter, as urbanization occurred, which means that infectious diseases are going to spread much more quickly. Not the question you asked, but That happened with the formation of towns and cities. So I think there's an interesting topic here. I'm not entirely certain what is the best diet for us. I am certain that we evolved in circumstances which had a different sort of diet to the one we have now, and I think we ought to understand it better, as I suspect you do too, so that we can have healthy living and longer lives. And we are maybe not doing that properly with the sorts of foods that are being sold, because we love eating fats, we love eating sugars, we love, including me, eating ice cream, and I'm not sure it's always so good for us.
Thank you very much. There's a hand at the two hands there. The one at the back came up first, so let's give...
First, but we're shifting to the left now.
Can we give a microphone to that young man? Is it young?
There's a gentleman in the front here with his hand up.
Yeah, there's one at the back there who put up the hand first, I think. Yes, okay.
Somebody speak.
Yeah, here we are. There's somebody there.
Hello. Yeah. I'm here.
Got you. Okay.
Thank you, Sir Paul. Amazing lecture. I'm an undergraduate student from University Malaya. Currently studying microbiology and molecular genetics. I'm Calvin. So I was thinking about quantum mutations, that the current technology that we have, that we are able to discover that electrons, they interact in a way that they can synchronize almost at the same time. And even there's quantum teleportations. And you mentioned about chemical structures in the DNA, or even the protein structure that is made of the chemical compounds. So is it possible that the movement of electrons could be one of the sources that contribute to genetic mutations, or possibly other modification in our genetic expressions?
Yeah. No, I didn't get the last part. Yeah.
I think the essence of his question is whether is it possible the electrons, is it electron movements?
Yeah.
Could be the source of some of the mutations.
Yeah. I think you're probably asking whether, could I generalize a little bit what you might be saying? Because I think maybe to focus only on electrons may get us into a smaller part. But what you might be saying is that the chemical nature of mutational change is not random. There can be more frequent chemical changes because of the nature of the chemical reactions. And I think we know that to be true, because we know that the changes, and I don't have them in my head which ones they are, but we're dealing with the 4 bases of DNA, and we know that they have a tendency to change in certain directions. Rather small differences, I think, but definitely there. So what that means is that will contribute to genetic change in a chemical way. And I think that's the question you're asking. And I think that that definitely will have an impact. And it is possible that it isn't just dependent on that particular molecule, but maybe influenced by the surrounding molecules. And if I can generalize a bit further, it's not just chemistry, because purely chemistry, because the chemistry can act as an intermediary through affecting molecules like DNA polymerase or RNA polymerase that are interacting with the DNA. So you could imagine that happening as well. So I think that the bottom line there is there will be a tendency, due to the chemistry, as you just say, to have certain more directed changes. I do not think that is going to end up with major directional change, but I think it could interact with small directional changes. And it is something I'm no expert about, but I know has been discussed and considered. So I think the answer is yes in principle, but it may not have a big impact.
Thank you, Sir
Thank
Paul.
you. There's a question there. I think it's going to move
One here, couple here. Yeah.
Hello. Hello. Hello, Sir Paul.
Hello.
Yeah, all right. I'm Tengku Muhammad. Okay. From Turing machine, we have mechanical computation. We have artificial neural network. We have.
Maybe speak clearer to your mic.
Could you
Even I
actually
have problem
put
hearing
it
you.
closer? Yeah.
Okay, I try again. Right.
That's better. Yeah.
Okay. From Turing machine, we have mechanical computation. We have mechanical thinking. Now we have artificial intelligence.
Yeah.
It is possible that we can build a super intelligent machine that is smarter than us.
Yes, so people talk, I got that. It's much better if you hold it close. It really is. Yes, people think about this. Now, the first thing I need to say is artificial intelligence. Can I say something about artificial intelligence? Because everybody is talking about it. Primarily, what we are seeing today is extremely sophisticated machine learning based on lots and lots of data and computational power, which allows one to predict outcomes that can happen, but without necessarily understanding how those outcomes come about. And the reason I emphasize it like that is that this aspect of artificial intelligence, I don't think is going to contribute very much to these sorts of problems of an artificially controlled machine yet. However, artificial intelligence is broader than machine learning, but I think it would be fair to say, and I suspect there's experts in the room beyond me, that the sort of artificial intelligence which is, if you like, thinking about how things would work, has not advanced very much in the last 50 years, including with our great increase in computational power. So I think the AI we have in place now will be very important, but I don't think addresses this question. But it doesn't mean that it may be, there may be a machine in the future that could address this question. I think that is absolutely not excluded, but I just don't think we are there. And by the way, I mean, people like Demis Hassabis, who is a deep mind person, completely agrees with that. He's a friend of mine. We talk about these things, and he does know what he's talking about, whereas I barely do in this area, at least. Now, then, thinking of designing a machine, I would say the first things that we're going to be seeing is beautifully controlled robotic extensions of things that we can do, which will deal with people who have been injured or maybe diseased, which we are already seeing the ability with brain implants and so on to get some sorts of control back. And that is intellectually, it's not a great leap in my view in terms of artificial intelligence, but it is an extension of the robot, if you like, so that we can actually do things better. But it is built on pre-existing life. Your question is, could we imagine designing a sophisticated life? I think, okay? And I think my answer is, I could imagine it, but I don't think it's going to happen anytime soon.
Thank you. I think there was a question there that had bypassed. If you can quickly get that question through.
Hello.
Oh, okay.
Hello.
The mic's with this person, but we got to get a mic to that gentleman there because he actually asked, he raised his hand much longer ago. Sorry about that.
Thank you. Thank you for the nice lecture. My question is this. You have defined what is life. I'm just wondering, how do you define death or demise of life?
How do you define death or demise of life? The death? how do you define death
Oh,
or the demise of life?
yes, beginnings and endings. Beginnings and endings. Difficult thing, isn't it? I mean, defining beginnings is morally and ethically very important because it influences what can be done with, for example, human embryos and what can be done in discovering how they work, which might help dealing with certain diseases, but which is, if you like, tampering with life in ways we have to be very, very careful about, in my view. At the moment, we've decided, but it's a bit arbitrary in many countries, it varies from country to country. I'm starting with life, I'll end the finish of life in a moment, but I think that, doing investigations in embryos which are no more than 2 weeks old is acceptable, but beyond that it becomes less acceptable. And that is largely driven, I think, by discussions about the nervous system and where that's being developed. Now, ending of life. We know that human beings can be kept alive without any brain function. But it's not life as we would think normal human life is. So the chemistry of life can be maintained, the body can be maintained, but the thought, the consciousness, as far as one can tell, cannot. So once again, we need the humanities to help us, in my view, and religion has views too on these matters as well. And it has varied over time. I mean, religious views of when something is alive or dead has changed over the centuries. But I suspect we will end up paying a lot of attention to consciousness and the sense of whether we still have a functioning brain, and that just keeping the body alive won't be sufficient, I suspect. But it is a really difficult one for medics. I mean, you should ask our question for my colleague here, because they have to make extremely difficult decisions in this situation, which are emotionally very challenging for them and for the families and friends of the individuals involved. And it isn't clear. It isn't clear. It certainly isn't just the ability to carry out the reactions of a human body, because that we could keep the body alive, but without brain function that suggests there's any consciousness left. So this is always going to be a muddy area. It's always got to be thought about. And frankly, we probably just have to consult with society and make decisions that people can live with. And they may change as beliefs change and knowledge changes. But key to this is getting the knowledge understood by society and then having it properly discussed. Because there's never going to be an exactly precise answer. And what makes it difficult is different cultures will have different solutions. And now we have more global societies, that increasingly matters. Sorry, I haven't answered your question.
Thank you. It's a difficult question, and it will embody it into area of definition of brain death and
for sure,
all kinds of issues, except to say that the responsibility for a good death depends on a good life. We have a question from here.
Hello, finally.
Yes, please.
Okay, my name is Sharil. I'm from.
Could you hold it really close?
Right. My name is Sharil. I'm from National Institute of Biotech, Malaysia. So I'm trained as a molecular biologist. I'm not going to ask you on consciousness, but I'm just going to be a bit more philosophical here. Many years ago, I read a book by Richard Dawkins. I think it's Selfish Genes, or probably I heard it from one of his talk, where he did mention that life is just how the DNA perpetuates itself, right? So I've been struggling with this concept. I wonder what your thoughts about it?
Yes, okay.
You said life is the ability to
No,
reproduce
how
itself.
the DNA,
Sorry?
how the DNA perpetuates itself.
How life, it's a question about how life perpetuates
No, the
itself.
DNA itself. How DNA took us as a vessel, to continue to reproduce itself.
I didn't quite catch you. So your question is, how did DNA manage to perpetuate itself?
No, I mean like how DNA hijack us, to perpetuate itself. It just said how the DNA, to actually to reproduce itself, rather than us having the concept of being alive.
I'm not quite sure I get you too, but your question, exactly as you say, is how did DNA hijack us to perpetuate itself?
Yeah, not really. So the concept is like this, where life is actually how DNA make itself living. Rather than
itself what?
Living, make itself living, perpetuates itself.
A living thing.
Yeah, rather than us having like a concept like a being alive, we become alive because the DNA wants to reproduce.
It's a question about how DNA brings about life.
Yes.
Yeah. Well, this is close to the major problem with the origin of life.
Yeah.
Because we can only imagine life when we have certain elements operative together. And one of them is, of course, the functioning of DNA as the hereditary material. The second is the building of a cell, which is a whole set of other things that need to be in place. And when we try and imagine putting all these different components together, we run into a problem because nothing will work without the other things working. And I think that's perhaps the nub of your problem. So I'll make one or two observations about it. The first is the distinction that I was making between DNA that is chemically inert but encoding for proteins that are chemically active. And that is the clever thing about life. Now, if you look at the molecule of RNA, RNA is actually able to encode information and to carry out poor catalysis. It is poor. And this has led to the concept, not mine, of what's said to be the RNA world, where the initial life might be built on RNA, and then from that, DNA and protein somehow was tagged on. So that's one of the concepts about origins of life. You make RNA with, chemically rich solutions that are made when there's lightning and things of this sort. So you could sort of, people think about the chemistry, but actually it's more the system, I think, which is actually the problem rather than the chemistry.
Yep.
I've given a bit of thought to individuals who propose that life arose in the vents in the middle,
Yeah, I've heard about it, yeah.
Now, the reason I gave some thought about that was that we need cells, ultimately, and yet you can't make cells, you have everything else. But if you have little holes in rocks, maybe they are the cells initially, and somehow the chemistry is made in these little cells, and if it's particularly effective, maybe RNA-based chemical, it invades into more little cells of the rocks and sort of dominates the system. And that might be a sort of stepping stone towards cells. But I think the nature of your question is really the nub of the why imagining how life formed is so difficult. And I want to say something else about that, if I may, which is often not fully appreciated. The solar system is 4.6 billion years old. The Earth formed a couple of 100 million years later, cooled down probably by 4 billion years to a form when life is possible. And then we have the first fossils only 300-400 million years later. Yet we take 3 billion years to go from the first cells to multicellular life with the Cambrian, Precambrian, Cambrian transition. Now, for me, making the cell is horrendously difficult, and it's the nub of your question.
Yeah.
Once you have a cell, I imagined you could make multicellular life, I mean, relatively easily. I don't see why it should take 3 billion years to do it.
Yeah.
Yet it seemed to happen in only a couple of 100 million years. And so I only add that as a, and I think you will probably want to say
Because-
something more, but I also think we're probably wanting to end. Is that correct?
All right. No, I mean, because, I believe that somehow people say that the origins of life starts as RNA, right? And then that's how it perpetuates to
about
become
RNA
DNA somehow, by the urine mill experiment where we have organic compounds and then it becomes a cell. So I thought that, it starts from RNA and it becomes DNA, and that's how it becomes us as a human.
But we do not know how life originated. We can only speculate, and not much more, which is why I never discuss it.
I think we should discuss this. I would have to use a chairman's prerogative there. There are two questions that came online, and there is a large audience online as well. One of which I think you more or less have answered to some extent. Let me try and recall this. And this question was from Anwar Azizan, who actually asked about the beginning of life, which I believe you have answered. And then your concept, your opinion about the minimum gene concept.
Minimum gene content. So this is a question people are quite interested in, which is, what is the lowest numbers of genes that are needed to give life? Now, normally this is thought about in cells which are living in the cells of other living organisms, so they can rely on many of the things of the other organism. And if you look at cells in that situation, you're down to about 500 genes, I think, something of that sort. Some people, of course, have claimed to have made life just by sequencing the whole DNA, but they cheated by putting it into pre-existing cells. But I think probably we need about 500 genes if you have a complex life support system. If you're looking at, I work with yeast, and that requires 5,000 genes. My lab has deleted every gene of those 5,000 genes. So, I mean, all that is possible. But for eukaryotes, which free are living, 4,000 or 5,000 genes would be the minimum. And bacteria are not much smaller. So for a proper free living organism, 4,000 or 5,000 genes, I'd say.
Thank you very much. So those trying to make a new life form, at least 500 genes. Okay. We actually have 1 minute left only, but this is a question from Cheryl Nizam. It says that you mentioned that to evolve is to grow and reproduce. What if one chooses not to reproduce? Are we considered not evolving?
If we don't reproduce, we will not evolve. And by the way, if we don't die, we won't evolve either. So with death, there is life.
Thank you. We're just on time, and I'm not going to use any time here to summarize this amazing session. It's impossible to summarize because it's so full of richness. But I'd like to end with just a quote from a poem, since you like poetry, I believe, Sir Paul, and so do I, from a Welsh poet who wrote about leisure, not about life. But he starts the poem with, "What is life? If full of care, you have no time to stand and stare." I think all of us here are not doing experiments at the moment. We're here very alive, but believing that we should be here to listen, to stand, to stare, to admire all that is being discussed. And I hope that summarizes how we feel about your lecture today. Thank you.
Thank you. Ladies and gentlemen, let's give another round of applause to Sir Paulus for the insightful sharing and Prof. Datuk Looi for guiding the discussion. Sir Paulus and Prof. Datuk Looi, you may return to your seats. Honorable guests, ladies and gentlemen, before we conclude today's event, we would like to take a moment to present a token of appreciation to the Nobel Laureate Sir Paulus. With utmost respect, we invite the Secretary General of MOSTI Yang Berbahagia, Datuk Technologist Dr. Haji Aminuddin bin Hasim, to join and witness the presentation of token honoring Sir Paulus by ASM and NIBM. Please welcome also Yang Berbahagia Academician Datuk Dr. Tengku Muhammad Azman Sharifuddin, Yang Berbahagia Professor Datuk Wira Dr. Raha binti Abdul Rahim to the front of the stage. We will also like to invite Sir Paulus, accompanied by Professor Datuk Looi, Mr. Jack Boyer, and Professor Evelyn Welsh. First, we would like to invite ASM President to present a token of appreciation. Next, we would like to invite CEO of NIBM to present the token of appreciation. Thank you, everyone. Please remain for a photography session. May I kindly request all honorable guests to kindly return to your seats? Ladies and gentlemen, with that, we conclude today's event. We invite all guests to join us for some light refreshments outside the hall. Thank you once again for your presence and participation.